Hi, my name's Erin Burns, and I'm a weed scientist and extension specialist at Michigan State University. And in this session, we're going to learn about identifying herbicide injury using herbicide sites of action. So why do we study herbicide modes and sites of action? And these two terms might seem something that you're not familiar with yet, but by the end of this session, we'll be pretty familiar with these two terms. And the first is that it's important for planning weed management programs. So we want to make sure we match the correct herbicide with the weed problem that we're trying to battle. Not all herbicides will control all weed species. So that helps us pair those two together. Also, which was what we'll focus largely today, is correctly diagnosing crop injury symptomology. And there are a number of color reference guides available. And at the very end of the session, I will provide some links that you can go and look on those later and help you use when you're out scouting in the field. And then we wanted to avoid the development of herbicide-resistant weeds. When we use the same herbicide over and over again, that speeds up the evolution of herbicide-resistant weeds, which means we can no longer use that herbicide to control that weed, which also leads problems with yield and ultimately economics on our farm. So I'm just going to go over two terms. And the first is herbicide mode of action. And the mode of action is the physiological or biochemical process within the plant that is impaired or inhibited by that herbicide, which is just a fancy way of saying how the herbicide works. So this is the biggest category of these two, the most general. Next is site of action. And this is the physical location within the plant that that herbicide actually goes in and binds. So this is where the herbicide works. And this is actually, we know most of these fairly well for our different herbicides, the exact location that they go in and bind. And this is what we'll focus on when we're diagnosing our crop injury symptomology. So to get an idea of how these two are nested into each other, first we have a herbicide mode of action. So how that herbicide works, that biggest category. And I just have one example on your screen. So the mode of action is amino acid synthesis inhibitors. Within that, we have two different sites of action. So this is where the herbicide binds, and those are ALS inhibitors and EPSP synthase inhibitors. So these bind in two different locations, but how they work is the same. So that's why they have the same mode of action. And then we have a number of herbicides within each site of action that share the same way of where they bind within that pathway within the plant. And I have just a number of examples on your screen in which the light red are ALS inhibitors. And the first term you see is the herbicide active ingredient. And in parentheses is the trade name. So that's the name that you would see on the herbicide label itself. So you might be more familiar with that terminology. And all of these herbicides, although they have different active ingredients, where those active ingredients bind within the plant are in the exact same space. So that's why they all share the same site of action. And you might be thinking, oh my gosh, I can't remember all these diverse site of action names. Well, they've numbered them for you, which is really great. So all we need to really remember is that different group numbers actually have different sites of action. So if they have two different numbers, you can know that where those herbicides work within the plant are different. So where do we find the site of action group number? And that is on the herbicide label. So on the majority of the herbicide labels, either on the central part of the label, which I have circled in red here. This is the Roundup Power Max label, which says group 9, and then herbicide, that is the site of action group number for Roundup, or sometimes it'll be on the top left or right, but it's usually right on the first page of that herbicide label. So that's pretty convenient when we're trying to look this up. So why do we use modes and sites of action? Once again, is to maintain a greater diversity in our herbicide use for herbicide resistance management. And that is because when we rotate among effective herbicide site of action groups, this will delay the development of herbicide resistant weeds. So if we use the same herbicide site of action over and over and over again, that is how we develop herbicide resistant weeds in our fields. Also then, if you do have a herbicide resistant weed, we want to make sure we're pairing the correct herbicide so that it's not in that same site of action so that we can actually control that weed. And then to diagnose crop injury symptomology, as often these are linked with the different herbicide modes and sites of action, which is what we're going to dive into for the rest of these slides. So why do we see herbicide symptomology on crops? And the first is normally or most common, is direct application of a herbicide to that crop, either by mistake or at the wrong rate or at the wrong application timing. Some herbicide or some crops can detoxify that herbicide when they're smaller, and then when they get larger, we might see some symptoms. So maybe we applied that herbicide at the wrong time. The second could be carryover application made from previous seasons. I'll point out some herbicide sites of action that stick around in the soil for a little bit longer than others. So there's a chance that the crop that you plant the following year could be injured from a herbicide that was actually applied the previous cropping season. And the third is drift or volatility from adjacent fields. So these are the three broadest categories of why we might see some crop injury symptoms. So how does that actually work? And herbicide selectivity is based on the crop's ability to rapidly metabolize the herbicide into non-toxic forms. So all that means is when the herbicide active ingredient is greater than that plant metabolism, than that detoxification rate, we see crop injury. And this can happen under a wide variety of environmental conditions, but usually when it's cold and wet, crops aren't growing as well, and that's often when we might see herbicide injury more often. So next, we're going to go over a few of the more common sites of action used across our cropping systems in the United States, and we're going to base this off of a classification chart, which you can see as a small snapshot on your screen. It's a pretty detailed chart, so we'll just go over a few of those. And you can actually go on our website at the end, and I'll give you that URL. It's the Take Action website, and you can get this poster sent to your house for free. So you could have this as a reference guide for you also. And this lists herbicides by a number of different categories. First, that mode of action, how that herbicide works, then the site of action group number, and also the site of action group name, and then the number of herbicide resistant weeds in the United States that we know about that are resistant to those certain sites of action, and then the actual chemical name, the active ingredient, and then the trade name, which is what you'd see on the label. Also, a large portion of this poster are premixes, which are herbicides that contain 2 or more active ingredients, and those are also color-coded for convenience when you're looking at the poster also. So this takes into account some of those too, so you can see what site of action might be in a premix that you're familiar with using. All right, so the first site of action or mode of action we're going to go over are lipid synthesis inhibitors. And the site of action is group number 1, which are ACCase inhibitors. These are selective, so that means that they control one group of plants and not the other. So dicots or broadleaf plants are tolerant to ACCase inhibitors because their ACCase enzyme does not bind these chemicals. So because of that, ACCase inhibitors control mostly grassy weeds. And our activity is mostly foliar, so applied to the foliage of the plant is when these are active. And then also there is some root absorption depending on the herbicide that we're talking about. Movement, they are translocated to areas of high lipid production. So this means apical meristems or roots. So you can think of just what the most active growing spots of that particular plant are often when we might see this injury because that's where this herbicide is moving to. And then key points is that it only kills grasses and it doesn't have any broadleaf activity. So this is a nice site of action that's fairly selective that helps us pare down that injury. And what's really nice is a picture on your right-hand screen. If you go up to a grass plant that's been injured by ACCase inhibitors, you can pull the center portion of that leaf, which we consider the whorl, which I have circled with the yellow circle on your screen, and then that will detach immediately from the plant. So that's a really nice symptomology that's just linked with this site of action. Injury symptoms usually appear within 7 to 14 days. And remember, they're usually only observed on grass plants, as you can see on the pictures on the right-hand side of your screen. And new leaf tissue will be yellow or brown. And then remember those leaves in the whorl, just that center growing point of the plant, will be easily separated from the plant. So if you had corn that was injured, you go up, pull the center portion of that plant out, and it would come out right into your hand. The second mode of action I'd like to discuss are amino acid synthesis inhibitors. And this mode of action has 2 different sites of action. The first we're going to discuss are group 2, which are ALS inhibitors. Selectivity for the ALS inhibitors is depending on the herbicide. Some can control grasses, some can control broadleaf weeds, and that's because they're used in a large variety of crops across the United States. Activity is foliar or root absorption, but this also depends on the herbicide. Movement, they are translocated, so they move throughout the plant system, once again to those growing points to the meristem or to the roots. And then some key points for ALS inhibitors. Some have very long soil residual activity, so they can have potential for carryover. So these are ones in which if you scouted this year, you'd want to know what was applied the previous season to know if an ALS inhibitor was applied and potential symptomology from that. And we do have many weeds that have evolved resistance to these herbicides as they are used so widely. Injury symptoms normally appear within 7 to 14 days, and above ground symptoms include stunting, so that plant stops growing, chlorosis, which is just a fancy term for yellowing of the leaf tissue, which you can see on the plant I'm circling now. That's what we could consider chlorosis. Also purpling of the leaves, which we'll see in the next slide, and then improper leaf unfurling. So you can see on these corn plants, those leaves that just don't look like they're unrolling correctly. And then also below ground symptoms can include a root development that's inhibited or pruned lateral roots, which sometimes we might get confused with some insect feeding. Sometimes it's hard to distinguish between the two, and what they consider this is bottle brush appearance, which is often from ALS herbicide carryover. This corn plant that I'm circling on your screen is an example of the root system that would be injured by an ALS inhibitor. Here are some more symptomology pictures. The top left is carryover, and then the purpling of both the veins and then the leaves of that corn plant in the bottom part of your screen. These are common symptomologies that we would link with ALS inhibitor injury. The second site of action that is in this same mode of action is group 9, which is EPSP synthase inhibitor, and the only member is glyphosate. And glyphosate is an active ingredient that's in the trade name Roundup, which you might be familiar with. Roundup is a widely used herbicide across the world, and it has many different formulations. So I haven't listed those all here. There's many different trade names for glyphosate, but the most common is Roundup. Selectivity is it's non-selective. So this means that anything that glyphosate or Roundup touches will be controlled. And this is the exception is if those crops were bred to be resistant to glyphosate, which is glyphosate resistant crops. So the picture on your right, we planted 2 rows of glyphosate resistant corn. Next to that, I have 2 rows of glyphosate sensitive corn, in which we then applied glyphosate over the top. And those sensitive crops will die because it's non-selective. So anything that is not genetically modified to be resistant to glyphosate will be controlled. It has only foliar absorption. Movement, it does translocate throughout the plant to those growing points. And then key points is it has no residual activity, so there's no carryover potential from glyphosate application. It is one of the most widely used herbicide, and because of that, we have a large number of glyphosate resistant weeds. Injury symptomology are usually a little bit longer than some of our other sites of action. It takes about 10 to 14 days to start to see glyphosate symptomology. Plant foliage, especially the new growth, will first turn yellow, and then it turns brown, and eventually death occurs. And this appears much more quickly on grasses than broadleaf plants, and grasses are highly susceptible to glyphosate injury. And also to note, we can see injury on some of our specialty crops. So I've included a picture of potatoes. The plant was applied, had a glyphosate injury, and then the tubers actually were malformed because that herbicide can translocate down to those growing points. So we can see some injury both on those also. All right, we're going to move on to the growth regulators. The site of action I'd like to discuss is group 4, which are plant growth regulators. These are selective, and they control mostly broadleaf weeds, including many perennials. Because they control broadleaves, we mostly use them in grass crops, but recently through the introduction of growth regulator tolerant or resistant soybeans, we start to see these this site of action used more widely outside of just corn production. And on the right-hand side of your screen is a picture of a weed showing group 4 injury symptomology, which we'll go over in a second, but this is a nice site of action too that has some distinctive symptomology that's not shared with some of the others. It does have foliar and some root absorption for activity. Movement is it's translocated throughout the plant, and that's why it can control many perennials, which we're targeting the root system of those perennials to get good control. And then key points is it mimics auxin type plant hormones, and there are some resistant weeds. Symptomology appears usually within 10 to 14 days, and broadleaf plants show stem twisting, which I'm circling right now, which is a hallmark of this site of action, callus tissue formation, so hardening of some of the plant tissue, and then leaf cupping. So you can see this soybean plant with cupped leaves. That's also a pretty good giveaway for group 4 injury symptomology. And then I did mention that mostly grass plants are tolerant, but we can see some injury to corn if we've applied some of our group 4 herbicides either at the wrong rate or when the corn plants are too tall. We will see leaves unrolling improperly, stalk bending or brittleness. You can actually go up and just snap that corn plant in half. Fused brace roots is just the picture I have on your screen right now. And if we got a big wind or storm event after that, we can see some lodging issues, and then also some missing kernels, which can have implications on yield. The next site of action we're going to go over are photosynthesis inhibitors. And photosynthesis inhibitors are unique to plants because plants photosynthesize. And plants are able to make their own food necessary for growth and reproduction because unlike you or I, they cannot eat. So they must make their own food to continue to grow. And to accomplish this, plants must utilize sunlight in a process known as photosynthesis. And photosynthesis transports sunlight, water, and carbon dioxide into a form of energy that the plant can actually utilize as food. And how they do this is the photosynthetic electron transport chain. And this is a great target for herbicides because that is unique to these plants. So we have a lot of different herbicides that will inhibit photosynthesis. The site of actions that I'm going to cover are photosystem 2 inhibitors. These are groups 5, 6, and 7. And they are very similar to each other, but where they bind within the photosynthetic electron transport chain is slightly different. So that is why they're assigned different group numbers. They have both pre-emergence and post-emergence activity. They are selective, but this really depends on the herbicide. So some can control grasses, some can control broadleaf weeds, and we use these sites of action in a variety of different crops. Movement for soil applied photosystem 2 inhibitors, they are translocated into the water conducting tissue, which we term xylem, which then I'll show you some symptomology that is linked to that movement. And fully applied photosystem 2 inhibitors are contact or non-translocated. So where that herbicide molecule or spray droplet touches the plant, that is where you'll start to see the symptomology first. It doesn't move throughout the plant like some of the other translocated sites of action that we've discussed. So for soil applied symptomology, injury symptoms usually follow the path of water movement, because remember those soil applied herbicides move in that xylem tissue, which is what the plant uses to move water about. And the symptoms usually appear in the older leaves first. So on the bottom right-hand side of your screen, you'll see some of those older leaves showing the symptomology, and the new growth can often be green. And then also nice for this particular site of action is termed interveinal chlorosis. So the veins actually remain green, but the spaces in between the veins are yellow or brown. So you can see this nicely on this photo in which these veins are green and then in between are yellow and brown. So that's a nice way to link this symptomology to this site of action. And then foliar applied symptomologies, remember these are contact herbicides, so they are not mobile. So injury symptomology usually appears first where the herbicide has come in contact with the plant. So bottom right-hand scene, we can see some speckling on these soybean plants. These were individual herbicide droplets of a photosystem 2 inhibitor that landed, and then that's where you see the symptomology. And then the corn plants in the top part of your screen, you can see only the older leaves are showing symptomology because those are the leaves that were out at the time of herbicide application, and then the newer leaves will be green. So remember, these herbicides don't translocate or move throughout the plant. So only the plant tissue that was there at that herbicide application will have the damage on it. Injury symptoms start as bronzing or speckling of plant tissue, and then it's followed by that general chlorosis and then necrosis, which just means plant death. And how quickly these injury symptoms appear is based both on the herbicide, what kind of additive, if we used any kind of adjuvants, and then the environment. So remember, these are inhibiting photosynthesis. So during sunny, bright conditions, when those plants are photosynthesizing very quickly, that is when we'll see oftentimes this herbicide injury appear faster under those conditions. It can be within hours oftentimes when we can see this symptomology. Next, we're going to go to pigment inhibitors. So the site of action I'd like to discuss is group 27, which are HPPD inhibitors. This is probably the more common site of action used within this mode of action. They are selective. They can control broadleaf weeds and some grasses, but this is largely herbicide dependent. We mostly use HPPD inhibitors in grass crops, but there are new resistant soybean crops that are going to become available in the future that will also be resistant to HPPD inhibitors. So we might see use become more popular with those introductions. They have activity both foliar and roots. They are translocated throughout the plant. And key points is they have distinct bleaching symptomology. You can see on the picture on the screen now, the foliage turns bright white, and we'll do this. And it's a pretty unique symptomology to this site of action. And there are potential for carryover possible. So this would be another one if you suspect that you would then ask what herbicide was applied the previous season. Symptoms usually show up within 10 to 14 days. Once again, the plants turn white or translucent. Sometimes we see chlorosis, but most of the time we see this bright bleaching, which you can see on most of the pictures on your screen right now, just how bright that foliage does turn. And then you can also see some plant stenting, but normally the bleaching symptom is what we link with group 27s. And then cell membrane disruptors. The site of action I'd like to discuss are group 14. These are PPO inhibitors. They mostly control broadleaf plants. However, certain PPOs do have activity on grasses. This is a large group once again. Most have post-emergence activity, but some do have pre-emergence activity also. And then they are mostly contact or non-translocated herbicides. PPO injury symptomology can occur quite quickly within 1 to 2 hours after exposure. Appearance first as water-soaked foliage, followed by general necrosis. And then environmental conditions favoring rapid plant growth, or if we use adjuvants such as crop oil concentrate, can increase the degree of injury. And these symptoms will appear most quickly under bright conditions or sunny conditions at application. The final group I'd like to discuss are seedling shoot growth inhibitors. The most common site of action used across the United States in this mode of action is group 15s, which are long chain fatty acid inhibitors. I've put just a few trade names up on your screen right now, things like Harness, Warrant, Dual, Outlook, Zidua. There are a large number of other group 15 herbicides. These are all just examples of long chain fatty acid inhibitors. They are selective, but this depends on the herbicide. And then the majority of group 15s are soil applied pre-emergence herbicide products that will inhibit the emergence and elongation of shoots of those weed seedlings that are germinating in the soil profile. And then they are translocated within that plant. Symptoms include stunted plants that do not fully emerge from the soil. Sometimes you can see leafing out underground, but many times grasses will emerge, but their shoots just don't unwrap properly. So you can see these corn plants that are showing group 15 injury. This is a nice symptomology linked to the site of action, in which we often see that those leaves simply just can't unwrap from the central part of the plant. And then soybeans may have crinkled or puckered leaves, and then a shortened midvein, which gives them this heart or drawstring appearance. So those are the pictures that I'm circling right now, in which we can see these soybean leaves that look like their midvein is being almost pulled down to the center, causing them to look like a heart shape. So that was a large number of information, but there are some really great resources out there in different keys. So there's a herbicide site of action key for injury symptoms. Also, 2 great publications that were put out for injury symptoms in soybean and then injury symptoms in corn. And then if you're interested in that poster that I used to kind of frame this session, you go to iwilltakeaction.com/weeds. You can request one of those for free and get one of those for your reference. And then also our msuweeds.com is our website that I share with the weed science team at Michigan State University. And we have lots of great information on there too that you can use to help you when you are out scouting for herbicide injury also. And with that, I've put my email up on the screen too. So if you have any questions moving forward, you can feel free to visit our website or shoot me an email. Thanks for listening.